Okay. Well, good morning, good afternoon, good evening, depending on where on the world you are. We're starting session 6 now of IWBDA on our final day. It's going to be a session about machine learning, and we have two great presenters for you this morning. First up, we are having a talk about improving predictability in biodesign using ensemble models by Bret Peterson. Bret Peterson is a computational project scientist at Lawrence Berkeley National Laboratory in the Quantitative Metabolic Modeling Group. And Bret, feel free to share the screen, your screen.
Sorry, I shared my screen before unmuting. That was a mistake. All right, thank you. Can you see my slides?
Yes.
Okay, thank you. All right, so I'm going to be talking about the automated recommendation tool, ART, that Tijana gave half of her keynote on. And I'm going to talk about a particular area we've been looking at a possibility of improving. So ART is really designed to be in both the sort of the learn and design part of the DBTL cycle. But if we can make very accurate predictions, then we're pretty confident that we can make good recommendations at that point. So that's it's the learn part that I will be focusing on today. So ART basically takes input of almost any form, so proteomics, transcriptomics, promoter combinations, and then for whatever output you are trying to optimize or target, it will make predictions on those values. And then those predictions, as I said, can be used to make recommendations. So Tijana showed this example. I think she showed several actual biological examples that ART has already been used for. This was 6 promoters on 6 genes for producing tryptophan with a GFP marker. And the nice thing, or the nice result they got was that not only did ART make predictions that ended up doing much better than the base strain, but they actually did better than any strain that had been tested thus far. So the way ART works is, it's made up of a bunch of regression models, and then each regression model is trained in sort of the standard way. And once you've trained it, then that model can make a prediction. So you would give it some point in the feature space, and it would make this. It would make give you a scalar value for that spot in in the regression space. So ART just uses regressor models that are already in the scikit-learn library. So these are the 7 that it currently uses. And then it uses one other regressor model, which is based on a teapot workflow. So teapot will sort of, before you create the model, it will do it will create a bunch of possible workflows, and then it uses genetic algorithms to sort of try to optimize this. So you can think of this as a pre-step before you start the training. But after the teapot workflow is gone, it's just like any of the other regressor models and gets trained the same way. And so all of those pieces are just sort of standard library regression models. The idea behind ART is that all these models, the reasons we have so many regressor models is they all have their own strengths and weaknesses. And the assumption is that those strengths and weaknesses may be somewhat independent. And so that by creating an ensemble, you may average out some of those errors and get a better prediction at the end. The other sort of important part of art is even though all these outputs are scalars, it creates an actual distribution of the prediction by training a distribution of weights for the models, and plus an additional error term. And so the way it does that is you break up the experimental data into here it's sort of k minus 1 sets, and then you have the last set is so all these sets are used to train these individual regression models, and then the last set is used to train this Bayesian inference on the weights and the noise level. And you can, since there's nothing special about this set over the other k-1 sets, you can do this repeatedly for each different one and continue to update these weights. It's sort of Bayesian, so your current belief is basically the weights are evenly distributed across the models in a Büecherl distribution, with some large sigma or standard deviation for the noise level. And then and that's your initial prior. And then as you do these trainings, you train a posterior, and then that becomes your prior for each additional step. So the way this actually looks is, so here's the sort of the Büecherl at the beginning. I'm only showing it for 3 models because it's hard to draw for more than 3 models, but it's in the center, so each one is sort of evenly weighted. And then this would be what the trained models output, and then again this noise term is just a Gaussian. So what is happening is you get these outputs from the model. You've trained this sort of weighting, but it's just whatever this weighting distribution is, it's still just a weighted average. So you're going to get some distribution that basically is bound by the largest and the smallest output of the models. And then that noise term is basically a convolution. So it's going to act as a smoothing function on that curve. And so if you're in a part of the space where you have had lots of examples, then you expect the models to produce similar outputs. So this will get condensed. And so this will, the weights will also be sort of create a condensed distribution. And hopefully whatever your test case is will agree with all the ones you've seen before. So that noise term will be small. And so you'll get a very narrow density here, so showing that our uncertainty is low. If you haven't seen it, if you're in a part of the feature space where you haven't been before, then the models are free to wander however they want. So you might get a large separation of these. So that will spread this out. And also your error term might be quite large. And so that can even extend it further. And so that's where how these densities basically show where we don't have a lot of information, or we don't trust the models to produce an accurate result, which is important for the recommendation part, because when you're in sort of the exploration mode, or you want to see where you have a lot of uncertainty, you might want to recommend models or recommend experiments where you aren't sure whether or not there's a good a solution there. So the experimentalists, they don't really care about much of this. They really want art to be just a black box. And their goal is, because once you get into multi-dimensional or high dimensional spaces, making predictions is pretty hard. And so they really just want to see what these recommendations are. But in order for that to work, we have to be able to be pretty confident that even if the topology of the underlying surface is challenging, that we still give a good result. So we want to make sure that our predictions are as robust as possible. So they don't care what if it's easy or hard, but we care. So we use this function as sort of an example of a hard type topology. The domain is basically 0 to 12, so basically 4 pi. So you're getting 2 peaks per dimension. So in 2D, you get these 4 peaks. And of course, the idea here is the problem is unlike this convex surface, if you get in the wrong area or if you get here and you just do gradient ascent, you're going to end up on the top of this small peak, but you'll never find this large peak. And one nice thing about using this function for testing is you can just make the problem harder by increasing the number of dimensions, because the number of peaks is basically 2 to the nth, right? So so that's sort of a nice feature of this function. And one thing we noticed when we started testing with these harder functions is that the teapot, which goes through that optimization of the workflow before you start training it, tends to dominate. And so so when it's not that hard, so 4 features, so now we're talking 16 peaks with one actual optimum, then the orange is basically showing how much of the weight is for the one teapot, and this green is for the weight that goes to the other 7 regressors. And you up that to 8, so now we have 256 peaks, and now the teapot is dominating even more. And at 16, where it's whatever it is, 65,536 peaks, it's really starting to do all the work itself, which is fine if it perfectly models it, but it means you're sort of losing the benefit of the ensemble, right? So this is that case where if you start relying on a single model, this weighting function basically does nothing. You're just multiplying and you get the single peak. And so all of what you're getting is basically whatever the error function is being convolved around this, the output of the model. So, which is shown by this. So the weight's all on one model. So since the teapot was the thing that was dominating, we thought, well, let's add additional teapots. And it turns out even just seeding the random number generator, because the teapot is super sensitive to the initial conditions, that you get very different workflows pretty easily. So they were pretty independent of each other. And then we sort of set up an experiment where we would just sort of a normal, we created training sets and test sets using a Latin hypercube sampling of the space. And then we would train art with different number of teapots in it and then see how it does. So again, this is our function. And the result we got, so these left columns are basically art as it is out there today. And so there's one teapot in it. And you can see as we add teapots, so this one has 2 teapots. So now the yellow is a new color, represents all the other teapots other than the one that has the largest weight. So the orange is still the teapot with the largest weight, but you see the second teapot has taken on a lot of the load. And when we go up to 4, the other 3 teapots are doing more of the load. And now as you make the problem harder, you see even as those other 7 regressors are kind of disappearing, the other teapots are not, and they are actually continue to carry load. And so you're getting the benefit of the ensemble as the problem gets harder. So this is just zoom in on that last one. the significance, the statistical significance is high for all of them, but this one actually has practical significance, and that the predictions you're getting are significantly or I should say are practically better, and that means here's just my quick simulation of R squared just to show that we're going from this range where the fit is not very great into this range where it really is following the curve better. And so that means that can be, these DBTL cycles can be quite long for us. Some of them are a few weeks per cycle. So making a better prediction can have a significant impact on the number of those cycles that you have to do. So for future work, we're actually integrating this into ART so that you'll have a parameter to change to set how many teapots you want as part of your ART model. And then we're going to start applying that to some of these ART's being used in a bunch of projects already, so we want to see how it does adding this feature in. And then we would like to investigate what these weight distributions actually mean. So if it's like shared between even if it's only a couple models that are carrying the load, then that's sort of the behavior we want or expect, where it's all this is true basically all the time, and so the density we get out is as good as we can do. It's possible that the models aren't always good at the same time, so that one model or one subset of models is good in some part of the feature space, and then another model or subset of models is good at another part of the feature. And then we might start seeing sort of bimodal densities that are coming from this sort of representation, right? And so we don't know if those actually occur in real experiments, but it's worth looking for those, because then we might not want to use, we might want to extend the weight space to include information from the feature, so that the weight space would be dependent on the feature space. And then finally, like how teapot's doing for the individual regressional models, we would like to start to automate the hyperparameter selection for ART, which might include like the number of teapot models to have in the model. And I'd just like to thank Hector Garcia Martin and Tijana Radivojevic for their help in this project. That's it.
Thank you very much for that. I don't see any questions yet in the Slack or the chat. Please feel free to type them in or raise your hand and ask them directly. In the meantime, I was wondering, so it seems like the ART recommends further experiments, but do they have to recommend, I guess, good experiments in quotes, or can it also recommend further experiments which will, I guess, push towards maybe the overall maximum rather than an individual maximum?
Yeah, you're absolutely right. So that's the real strength of getting probability density out, is that we call it, there's a function that there's an alpha term that basically you set whether you're in sort of the exploration phase or the exploitation phase. And when you're in exploration, you basically use that uncertainty measure to show where there's where there's a lot of uncertainty, that is where you might make recommendations for more experiments. And then once you're fairly certain that you know the space, then you go into the exploitation mode, and then it focuses on just optimization.
Thank you. And Gonzalo asked, would you mind explaining what teapot is again, please?
Yeah, it's called the, let's see, tree-based pipeline optimization tool. So if you think of like a lot of things that you might do by hand if you were starting to designing a new regression model for a bunch of data, it tries to automate those things, or it goes through a bunch of possible automatizations and creates a tree workflow to create those. So you might like normalize feature space so that the dimensions are comparable. You might do feature reduction, like principal components, that kind of stuff. And then you actually select which regressor you're going to use. And then I'm trying to think on the outs. Anyway, so those kinds of things that you would normally do are all have been sort of compartmentalized into modules, and they create sort of a bunch of random workflow trees, and then whichever ones seem to produce the best results for the data set that you give it, it will basically duplicate those and then start swapping parts, so that's the sort of the genetic algorithm parts. And they keep doing that for hundreds of cycles until they get an optimum.
I hope that answers your question, Gonzalo. I'm going to ask one more question and then move on to the next one because we're a little behind. Michael asked, now that you've identified teapot as the model that gets selected most, will you eliminate the rest of the ensemble?
We did it both ways. So originally I did it, I basically swapped teapot. When I added teapots, I basically replaced some of the other ones. But I think it's safer because there's nothing to lose by leaving in more models. Because when the weights are learned through those Bayesian updates, if a model is not performing well, the weights will go to 0 anyway. So we have nothing to lose by it, and in some cases they may add, so we will leave them in.
Okay, thank you very much for your talk. Next we have Lukas presenting the comparison of intrinsic noise model predictions for genetic circuit failures. Lukas Büecherl is part of the Genetic Logic Lab at CU Boulder and is a PhD student in biomedical engineering, working towards a certificate in interdisciplinary quantitative biology.
Thank you. Oh, sorry. Yeah, thank you, Jet, for the introduction. As Jet said, I want to talk about extrinsic and intrinsic noise modeling for genetic circuits and the influence on genetic circuit failures. I want to start with a little bit background, a little backstory. So let's think about, so my background is in electrical engineering. I started with synthetic biology when I joined the Genetic Logic Lab here at CU Boulder. And let's assume that starting new synthetic biology would be a nice thing to join the iGEM competition. And in a fiction team like the IWBDA iGEM team, for example, we want to build a reporter that does some GFB production for certain input combinations. So how we would tackle the problem, or how I would tackle the problem, is we kind of think about when I want the reporter to be on, depending on sensor inputs that we have for the circuit. Of course, being an electrical engineer, I don't have a lot of background in biology. So it's kind of would be the most difficult part to get to a genetic circuit which we basically can just put together and put into a cell. Quick note here, like as standard, 0 means no output or no production, and 1 would mean we have a high output or production of YFP in our case. Luckily, I don't need to go in the nitty-gritty and kind of assemble the DNA myself, because I wouldn't even say recently anymore, but the last few years, genetic design automation tools became more and more standard in synthetic biology. So in our case, we would be able to define our logic table in the Verilog language and could use Cello, which is a software tool that was developed by Nielsen et al. And published on in 2016, and could use this tool to get our mapped DNA sequence, and we would have our genetic circuit. Sounds very simple. So the next step, how we would tackle this problem, would probably be that we order our DNA parts, go into the wet lab, do Gibson assembly, biobrick assembly, whatever assembly we feel like doing, assemble the circuit, put it into a cell, and test it. So we would look at the circuit in our cells, we would apply different input combinations to see if they do what we want them to do. And then if we look for all the input transitions here on the right with a green graph, we would see, yeah, they're supposed to be on, they are on, so that's working awesome. We look at the purple-ish color graphs and be like, okay, they're off, we want them to be off, so that's working. But then suddenly we would come to like one input transition that's supposed to be on, in our case, the transition from 0, 0, 0 to 1, 1, 0. And we'll be like a little bit confused because it's supposed to be on the whole time, but it would turn off and then on again. And that would probably be the most frustrating part of the project, because it would go over and over again if we like used Cello correctly, if we mixed up something with the state space somewhere. And in the end, it would be really frustrating because we couldn't find an arrow. However, we would still kind of trying to find out where it comes from. And luckily, in my lab, my colleague Pedro Fontanarrosa published a paper on exactly that problem and presented that, if I'm correctly, 2 years ago at IWBDA. And what my colleague was looking at is, where are those glitches coming from? So glitching is an unwanted system variation, and it's basically what I just described, that the output doesn't behave the way we want it to behave. If we look at this genetic circuit that we have here, we notice that we have an asynchronous circuit, meaning the gates are basically updated on the fly, meaning if the input transition of a gate changes, the output transition will change. So if we look at the p-tag promoter, which is basically the first input here, it's only one gate before we reach the final OR gate, the YFP gate. However, if we look at p-bet or the p-tag promoter, we have multiple levels of logic, which kind of makes it clear that the p-tag promoter is faster in influencing the output, which causes a switching variation. So let's take a look at that. We said we start with 0, 0, 0. So that's what we do here. Everything is 0. Everything gets updated on the fly. We get YFP production. Very nice. So now we change to our 1, 1, 0 input here, and we can see that the p-tag path is faster than the other path. So YFP gets turned on for a second till the p-bet promoter basically is fast enough to update their gates to then turn YFP on again. So that is why we see this glitch. There are similar problems in electronic circuit design, in asynchronous electronic circuit design. So usually what you do in electrical engineering, you use synchronous circuits, meaning you have a clock, and the speed of the clock is basically your longest or critical path. And with that, you just basically update every time. When the clock hits like this time, you update your states, and then you don't get this glitching behavior, which has the advantage that you don't see this glitching behavior, but the disadvantage that it's obviously slower because you always take the longest critical path. So now if you think about it, if we just think about our imaginary iGEM team, it's probably not too bad if we have like this glitch. It probably isn't too important. Now think about, you actually have a thesis that is based on something that is like actually really important, and the output is not YFP production, but maybe apoptosis or a drug release, then irreversible changes, these glitches, can have drastic consequences. For example, if your drug is released on an off-target tissue where it is toxic, or you want to control the death of your cells using apoptosis, but then suddenly your cells die when you do not want them to die. So for the safe operation of a genetic circuit, we kind of want to avoid these unwanted variations in the output. Okay, so here I want to take a little break and talk about what we do in our lab. So as mentioned before, the work of my colleague Pedro Fontanarrosa that was presented at the IWBDA 2 years ago was investigating why these glitches are happening. Then in the work that I was presenting at last IWBDA, we were looking at the probability of those glitches occurring. And in this year's IWBDA, we want to talk about the influence of noise on the circuit failures. So we have different sources of noise that can generate the variability in the circuit's output, and noise that we look at, we divide it basically in intrinsic and extrinsic noise. Yeah, and noise can obviously increase the probability of a circuit failure. So before we go to the results in a second, I kind of want to take a minute to talk about intrinsic and extrinsic noise. Intrinsic noise is due to the just inherent stochasticity of biochemical processes. As an example, when we use a gene regulatory circuit like we did here, we have transcription and translation, and it's never exactly specified, for example, how fast the RNA polymerase transcribes the DNA to mRNA, or how many ribosomes are there that are available for translation right now, how the resources are in the cell, are there enough amino acids. Some RNAs might be useless because the terminator doesn't do its job and it runs over, so they can't be used. We have just all these factors that kind of influence and make it harder to predict how these biochemical processes exactly take place. Luckily, we can look at them and simulate those using the master equations and stochastic simulation algorithms. So in our work, we use the Gillespie algorithm. For extrinsic noise, we have variations between cells and their environments, and the cell-to-cell variability manifests as differences in each cell's reaction rates, and it's kind of due to the influence of the environment. We can also analyze and simulate those. What we do here, we run an ODE simulation. So we don't run a stochastic simulation, but an ODE simulation. But for each run, we draw the parameter from a random distribution. So for example, for the degradation rates, or in general, the reaction rates, we draw from a random distribution. And I mean, the mean value is the value that we have in the literature for that reaction, but then we basically build a distribution around it, and from this distribution we take our parameter value. And Jake Beal argues that that distribution is a log normal distribution. So what are we aiming for in our work? We kind of hope that we can find differences depending on what modeling we use, intrinsic or extrinsic, and how that influences the probability of the circuit failure. For that, we look at 3 different circuits, which I will show in a minute, and our different noise models that I just mentioned. So the circuits we are looking at is the original circuit, or the original design that I showed earlier, which is circuit 0x8E, which is one of the 60 circuits that was originally published in the Cello paper by Newsnet on in 2016. And my colleague Pedro Fontanarrosa in his work from 2 years ago, he redesigned the circuit and came up with 2 new versions, the redundant logic design and logic has a free design. So if we look at the redundant logic design, the only thing you'll notice is different is that we have 2 inverters in the IPTG pathway, which basically just adds extra delay. I mean, 2 inverters do nothing. But they add extra delay, and the idea was that that helps with the glitching behavior that we saw earlier. We also have a logic hazard free design. For that, I want to kind of take a step back. So far, we've only been looking at function hazards. Function hazards are due to the circuit's function and therefore unavoidable. So if we look at these three circuit designs, they all implement the same function, the same truth table. And function hazards are due to the function, and these circuits all have the same function, so they all have the hazards for these input transitions that we look at. However, there are also hazards that are due to the logic of a circuit, which is basically the layout of a circuit. And if you use certain hazard preservation optimization techniques, you kind of can get away with no logic hazards, but the function hazards will still remain. Okay. So if we now look at our designs, here we have the 12 input transitions that we were looking at, and we have a probability for the glitching behavior. It's not, oh, excuse me. So it's not too important about the numbers or probabilities. What's more important about the intrinsic noise modeling is that we can see that the redundant logic design and the logic hazard-free design do better than the original design, which is shown by the green shaded areas. Those are the ones that do best for the input transition. And we can see logic hazard-free and redundant logic has wins in 4 transitions, and the original design is only decent in 1. If we compare that with the extrinsic noise modeling, the logic hazard-free one is the clear winner, and the redundant logic one and the original design are kind of on the same level. But it becomes the most interesting if we kind of take the one table and the other table and put them over each other, which gives this table. And here we see that the green shaded area with the cross in it, those are where the intrinsic and extrinsic noise model gave the same prediction. And we can see that the logic hazard-free and the redundant logic one are clearly better than the original design and win in most cases. So what kind of conclusions can we draw from that? First of all, the redundant logic and the logic hazard-free circuits are the better choice, usually performing better than the original design. Also, always an important point in this glitching behavior analysis is that it's important what input transitions the designer is interested in. What we mean by that is if we look here, and for our reporter, we mainly use the first 2 and the last 2 input transitions, then obviously we would choose a logic hazard-free design. If we're more interested about all the transitions we have in the middle there, then we would probably choose the redundant logic design. So there's also a very important point, what input transitions are actually needed. But it's also interesting that these conclusions are independent on what noise modeling we choose, because both for the intrinsic and the extrinsic noise modeling, the redundant logic and logic hazard-free one outperform the original design. So that is kind of where we are right now. We have a lot of idea for future work. First of all, it would be awesome if we could characterize gate parameters. Right now we have abliessence default parameters, but actually getting parameters like from checking the gates in the wet lab would increase the accuracy and be super interesting to see how close they are actually. We've also we've only looking at function hazards. I meant that briefly earlier. There are a lot of different other hazards that we kind of want to look at. So here are some mentioned, for example, the whole state, where you basically put the circuit in a state and we test for the ability how long it is able to maintain that state, or when does it flip without it should flip. Setup glitches, which are just glitches that happen when you initialize the circuit. Also important because if you initialize the circuit and then all your cells are dead because there's a glitch, it's also very frustrating. And then we want to work on our extrinsic noise modeling to extend it to use the model proposed by Jake Beadle. And finally, I mean, the coolest thing would be if we have a model that simulates both intrinsic and extrinsic sources of noise at the same time. So that's where we overall hope to go. And then finally, for the future, it'd be awesome to actually check all of that in the wet lab and see how our predictions actually compare to results in vivo. With that, I want to acknowledge the University of Utah, where my colleague Pedro Fontanarrosa is working, University of Colorado Boulder, where I'm working, and the funds for this research, the National Science Foundation and DARPA, and of course the Genetic Logic Lab under our PI, Professor Chris Myers, and everyone else in our lab was really helpful with our work. With that, I say thank you, and I'm open for questions. I will open the videos again so I can see you all, and thanks for listening.
Thank you for that talk. I don't see any questions yet, but again, please put them in either the Slack channel or the messages. Oh,
I think Jake Büecherl raised his hand. I assume that's the question.
Yeah, I was wondering, I'm surprised to see the intrinsic noise having such a large effect. Can you say anything about how you set the magnitude of the noises? Because my experience with strong circuits like the ones that cello has, that intrinsic ought to be very small because the molecule counts are large.
Yes, that is a very good point, and I hope I can give a satisfying answer. So it was a really good point that we got from a reviewer from our abstract. So far we use as an input signal 60 molecules, but obviously in a cell, the input is something like 10 to the power of 5, 10, something like that. It's definitely not 60 molecules that you have as an input in a cell. So that is something that we need to update to make it more a little bit real world like. We got to those probabilities by using constraints, saying, I did an analysis of a NOT gate, just a simple NOT gate, and I kind of did an input sweep to see how much input change I need to trigger the output. And based on that, I defined two values. So the input is 60 molecules, and I set 0 glitches when we drop below 10, and a high glitches when we go above 30. The 10 and 30 I got from my input sweep from the NOT gate. That's what I used as a threshold. And then I run my stochastic simulation. Every time I went over the constraint, the run was stopped, and then I had the probability of how likely it is that it glitches.
So if I'm understanding correctly, because you're wanting to really understand where a glitch can happen, you're basically turning up the noise until it's high enough that you can produce a glitch.
I wonder if you can say it that way, but yes. So I think what's definitely correct is if we use bigger, also higher molecular counts, then the influence of intrinsic noise should decrease. I think that is what we thought as well, which is something that we hope to explore in future work. But it's a really good argument, absolutely.
Thank you.
I also had a question about the general automation of the pipeline. How doable is it for someone who has their own circuit to do this kind of analysis at the moment, to replicate your analysis? So
there are two main ways you can do what we did. So probably the easiest way is to go to the GitHub repository and download the SPO and SBML files or the combined archive, open up Biosim and just run the simulation. That should work. If you want to do it by hand with a different circuit, you basically we use the Biosim application. So you open up Biosim, you build your circuit in ASPL, you can annotate it with real life parts, and then run the simulation in a Biosim. It should be reproducible. I mean, I hope so. But I think the easiest way is just getting the project which is available and rerun the analysis.
Okay, if there are no more questions, I would like to just point out that there's a break social now in GatherTown, where of course you can ask more questions to our presenters this morning.